Good evening. I'd like to welcome everybody to the University of Washington Molecular Medicine Public Lecture. Our lecturer tonight is Dr. Brad Cookson from the Department of Laboratory Medicine, and I'm the director of the Molecular Medicine Training Program. This is a graduate program that brings elements of medical training to our PhD students, and we're lucky to have funding from this program from the Howard Hughes Medical Institute that enables us to put on what turns out to be a very, very popular lecture series. So I'd like to welcome you briefly and tell you that tonight's lecture is by Professor Brad Cookson, who is in the Department of Laboratory Medicine at the University of Washington. Brad is from Utah. His father, he tells me, was literally a rocket scientist. And Brad went on to get his MD PhD degree at Washington University in St. Louis, and then couldn't resist coming to the West again, where he has been here in Seattle for many years. We won't disclose the number. And we have heard some wonderful reports about Dr. Cookson's research and about Dr. Cookson's ability to communicate his enthusiasm for research and science and all kinds of things. And I know our high school students visiting from Eastlake High School in Sammamish, is that right, who we're very glad to have here tonight, have gotten to enjoy a dinner with Dr. Cookson, and now we get to hear what he says. And the title is The Difference Between Medicine and Poison is the Dose, which is a very intriguing lecture title. So, Brad?
It is indeed a privilege for me to be here this evening to address you and to describe some of the research which you all have paid for. Okay, inflammation is the topic this evening, and it's described to be a secret killer. Hopefully, after this evening's lecture, there will be fewer secrets to all of you. Physiologically, inflammation is literally a pit bull. And it can cause horrible disease, but just like a pit bull, it also has its attributes. And one of the most important things for you to appreciate is that inflammation is also your friend. So classically, inflammation was defined in the 1st century AD. These are Latin words: rubor or redness, tumor or swelling, calor or warmth, dolor or pain. And Brad Cookson added the 5th, which is loss of function. And so inflammation is a property that's shared by 4 leading causes of morbidity and mortality in the developing world. And one of the reasons I thanked you 2 slides ago is that I prefer the Socratic method. And so to fill out the rest of this slide, I invite audience participation. In our country, 4 of the leading causes of morbidity and mortality are heart disease. Obesity fits in with heart disease. I heard cancer, stroke, and I heard HIV, which is infection. So congratulations, you hit all 4. So these major causes of morbidity and mortality are all linked. They have a commonality in inflammation or inappropriate responses to inflammation. And one of the problems with inflammation is that it's a big word. It's not a big word in that it's hard to say or spell, but it's broadly applied. And in its broad application, it can lose meaning. So, for example, if you compare, if we mix together coal and diamonds and a carbon fiber bicycle frame, they're all based in carbon, but if you swirl them all together, trying to ascertain their attributes would be extremely difficult. So, by example, let me touch upon one very common, very big word, which is drug. So when I was in medical school in the 80s, there was this campaign of to just say no. And this is Nancy Reagan in 1986 and her Just Say No campaign. And she wanted you to just say no to drugs. And as a medical student, my question was, which ones? Aspirin, it wouldn't meet FDA approval today, but aspirin has some very useful properties in altering platelet function that protect us from thromboembolic events. What about penicillin? It's also a drug, and its administration can provide life-saving benefit. And then there's a newer class of drugs, for example, insulin, which is really a peptide hormone, but when produced in a recombinant form in a biotechnology lab, it is also a drug. So my question then is, who really cares? Well, the reason I suggest that we care is that if we understand the cause of inflammation, the reason we care is because inflammation is linked between or connects 4 of the leading causes of suffering in our country. And if we understand mechanism or understand cause, we have the opportunity to improve diagnosis, treatment, as well as potentially prevention. Now it's interesting, I graduated from college in 1983, and the reason I chose medicine and research as a career is for this reason. By understanding mechanism, potentially we can improve diagnoses, treatment, and ultimately we hope to prevent disease and prolong life and improve the outcome for patients. And some quarter of a century later, those 7 words still summarize my professional aspirations. So this evening I want to put inflammation into context. And as Nancy mentioned, I'm a microbiologist, so by definition, I'm going to have a microbiological bias to my interpretation of inflammation. But I hope to solidify or fortify my points by also including historical references. So as a physician, I took the Hippocratic Oath, which was to prescribe regimens for the good of my patients according to my ability and my judgment, and never to do harm to anyone. And so if we consider Hippocrates to be the father of modern medicine, we can consider Sir William Osler to be the father of medical education, because he insisted upon medical students actually learning by being present at the bedside. And so what did he say back in 1896? Humanity has but 3 great enemies: fever, famine, and war. Of these, by far the greatest, by far the most terrible, is fever. Now, it turns out in the 1800s, there were other scientists making important discoveries that puts this terrible enemy of fever into context. And those were discoveries by Louis Pasteur and Robert Koch that began to discover microbes as the causative agents of infection. And then furthermore, later in the 1880s, Hans Christian Graham developed the Graham's stain, which allowed for differential staining of microorganisms compared to host cells. And some 200 years earlier, the single-lens microscope was developed. So now I'd like to give you an appreciation, sort of a rocket ship's ride through history. Imagine, as a physician, you're looking at what might be considered a fried egg sandwich. Actually, that's a sputum specimen that's brought from the lungs up through the major airways into the oropharynx and out the oral cavity. And if you get up very close to this, you might be able to appreciate something that looks like that. But in terms of defining etiology, we're millions of metrics away from being able to assign what's going on there. So in combination with using the Graham stain and microscopy, this can be converted into something that's much more beautiful, particularly if you're a microbiologist. And what is shown here, and now we're working on the micron scale, and microns, as you recall, are 1/millionth of a meter. So these purple round forms, or cocci, are bacteria. And near them, this sort of pink staining, multi-lobed entities are host cells. These are phagocytes, and phagocytes comes from the Latin roots of to eat. And so the phagocytes have emerged into a site of infection because these bacteria are unwanted. So this puts into perspective, potentially, what the fever is here that Sir William Osler so greatly feared. And now I have to ask you, is there anyone in this room that has ideas about our progress in these areas with respect to famine? Okay, we've improved farming practices, water conservation, fertilizer, transport. We still don't have famine licked, but we've made great progress. With respect to war, a very hot topic, okay, we've improved our defense against that, let's say, by having better body armor. So one of the fellows in our program, Lieutenant Colonel Steve Malin, spent a year in Iraq before he joined our clinical fellowship program. And it's remarkable how the destruction war has actually changed because of improved body armor. And so how have we addressed this concept of fever and improving patient lives? Well, I'm sure some of you are thinking, where did this clown go to medical school? Has he not heard of antibiotics? I have. The question I have for you is, how many people in this room have relatives that were born before 1945? That's a trick question, and I got almost everyone to answer the question correctly, which is, we all do. And so what is the significance of that? Is that many of our relatives existed pre-discovery of antibiotics. And so they were discovered by Alexander Fleming in 1928 and found to rescue infected mice in 1940. And then the first human recipient, which was a police officer in London, received penicillin, and it helped him until they ran out of penicillin because they had difficulty purifying it. And actually, the process of harvesting penicillin was the work of Chain and Florey, who shared the Nobel Prize in 1945 for the discovery of penicillin. And the question is, was this of significance? This literally brought the United States into the area of pharmaceuticals. And the reason is, and you can see here that in 1942 we had sufficient quantities to treat patients, but not until 1945 did we have non-military use of antibiotics. And the reason is because in World War II, we learned something from World War I, which was battlefield mortality due to infection exceeded 50%. And so the Allied forces thought that by improving the ability to make penicillin, that would be a huge benefit to our soldiers. Now, that stands then as a historical reference to the significance of infection in taking people's lives. Obviously, there are people born before this. My grandmother was born before the automobile and the airplane and the antibiotics, and she lived through the era of automobiles, airplanes, and antibiotics. It was also interesting that she practiced Christian Science, so she never had a pill in her life, never had a vaccine, and lived to the ripe age of 83. That stands out as a tribute to a system that must have been able to distinguish foreign from self. And I think it's reasonable, given the introduction I've given you, for you to say that, well, that's not so hard. You just showed us a gram stain, and here you can see that the morphology of these bacteria are very different than the morphology of these host phagocytes. And that's true. So my question is, does it sound too simple? And for that, I need a volunteer. Could I have one volunteer please come down? May I please have one volunteer come down? Please come down. Thank you very much. Now, biologically, we as humans, thank you very much. Could you just stand right here, and I'm going to ask you to assume anatomic position, which is like this. Thank you very much. Now, as biological entities, we're very complicated. And so for 1 minute, let's consider this person from the microbiological perspective. So I have a couple of questions. Did you eat today?
Yes.
Good. So she acquired nutrition. Did you have to move from the place where you slept last night in order to acquire that nutrition?
Yes.
Perfect. Okay. So we have a mobile entity that acquires nutrients. And may I ask, is your temperature above room temperature, boiling, somewhere in between?
I would guess it's above room temperature.
That's correct. So, from a microbiological perspective, we have discovered a temperature-controlled bag of nutrients that is supported by an endoskeleton. Okay. Now let's then consider if we go from head to toe, and we're going to take those infinitely small slices dx that we learned in the fundamental theorem of the calculus, and we're going to count every single cell in this individual's body. Would we get an accurate assessment of her cells by counting every single cell in her body, every single cell in this space? The answer is we would not. The reason is that we would have to distinguish between her cells and the other cells associated with her, which are other microbes. Okay? And so if we do this differential counting from head to toe, it turns out we find that she's outnumbered 10 to 1 by microorganisms. So you, in fact, share this space. You're a cellular minority in space predominated by bacteria. And so this brings forth the concept of microbiota. Are these microbes important to her life? The answer is definitively yes. For digesting certain types of nutrients, for acquiring certain types of vitamins, for example, vitamin K that's critical for coagulation, we rely on our normal microbiota. So I would like to thank you for your very capable help. This then suggests that there's a coevolutionary dynamic between the host and the bacteria that are present. And so that means that over time, pre-antibiotics, since we've lived for millions of years, there must be a system not to just distinguish between foreign and self, but to distinguish unwanted foreign from self. And how can we distinguish unwanted foreign from self? My capable assistant was not febrile. She did not have a fever. She was not suffering inflammation. So, inflammation, I would argue, is the result of our bodies or a system within us that is capable of interpreting danger signals. And these danger signals are those interpreted by the immunological self-defense system. And this system is responsible for inflammation. And throughout human development, human evolution, we had to develop a system to help us deal with infection. Now, how do we put this in context with the 4 leading causes of morbidity and mortality? Long before we suffered heart disease, long before we suffered stroke or from cancer, we were dying like flies because of infection. Infection has played a major role in almost all of the major human conflicts. And so, at this point, it should be clear to all of you that microbiology is a field of historical and global significance, and it is obviously the career of choice for bright young scientists. The second important point that I have is that the system responsible for causing inflammation evolved to deal with microbial pathogens, and because we can genetically modify pathogens, they're manipulable, it provides us with an opportunity to study the process of inflammation that is so important to these major causes of suffering of humans. And so with that, we leave part number 1. Microbes as a tool to study inflammation. Logic clearly dictates that the needs of the many outweigh the needs of the few. Mr. Spock, 1982. Imagine an altruistic system where one cell sounds an alarm because something bad is happening, because in my capable assistant, she is actually composed of a series of organ systems which are composed of specialized tissues, and those tissues are made up of individual cells. And so that for one cell to sound the alarm to protect the tissue and the organ protects the organism. Hence the significance of inflammation in our lives. And I point out the good part of inflammation. So this is how we conceived of it. The screaming, alarm-ringing, pro-inflammatory death of a potentially dangerous cell in an organism where prompt recruitment of additional cells or cellular functions are necessary to promote a positive outcome for the host. So, the setting has been laid out. Microbes are a great tool for investigating the process of inflammation, which has manifestations in other clinical diseases. Now let's bring in the scientists, 3 really capable graduate students. Brennan, first name Molly, now Molly Bergman, started much of this work. Currently, she's trying to decide which faculty position to take. She just completed her postdoctoral studies at Tufts University with Ralph Isberg. 2 other capable students. Susan Leilani Fink has just finished her medical training. She's off to Yale in July to do her residency in laboratory medicine. And Tessa Bergsbakken also recently finished and is doing her postdoc with Mike Bevan in the Howard Hughes Medical Institute here at the UW. A system we chose to probe the inflammatory process is salmonellosis. Now, you've probably heard about Salmonella this year. There was a big peanut butter outbreak. Last year, it was jalapeno peppers. This organism has evolved to cause disease in a variety of hosts. It causes localized as well as systemic inflammatory conditions. The important points here are that this organism has evolved to cause disease in a wide variety of hosts. It causes inflammatory conditions, but also importantly, it causes chronic infection. Sir William Osler also told us that if you listen carefully to the patient, they will tell you their diagnosis. Chronic infection provides for us an indication that this microorganism has evolved a way to escape immune detection, to live in the absence of inflammation by avoiding it. This suggests then that this is a potent tool for probing the mechanisms of inflammation. So, as I showed to you in the Gram stain, bacteria often associate with host cells. Salmonella is a rod-shaped organism that you can see here on this scanning electron micrograph. The scale is, this hot dog is about 2 to 4 microns long by 1 to 2 microns wide. And for this bacterium to cause disease in so many different hosts, it interacts with the phagocytes that I showed you on the previous Gram stain. For this bacterium to cause disease, it has to interact with phagocytes. It transports them from the GI tract to the mucosa to other systemic sites. The organism actually grows inside of phagocytes. And so for these aspects of the infection, the organism actually associates with the host cell that's designed to destroy it. And so seemingly at odds with this interaction results in cell death. And the important point I will tell you in advance is that this is a host-driven process that is defensive in nature. So what did we know about cell death? We knew that DNA was damaged in the host cells infected by this bacterium, and it depended upon a particular protease called caspase 1. And a protease is a protein that goes around and clips other proteins. And some of the proteins that it clips include IL-1 beta. And when IL-1 beta is cleaved into its mature form and secreted, it becomes what has been described through a number of decades of research as endogenous pyrogens. IL-1 beta is responsible for causing fever. And this process then of host cell death and the liberation or activation of IL-1 beta embodies the process that we call inflammation. This could also be described as caspase-1 dependent pro-inflammatory programmed cell death. It's a program because there are a number of biochemical steps. The cell dies because it's no longer capable of surviving. It depends upon this caspase or protease that clips other proteins, and the end result is that it's inflammatory. But that's entirely too long, and I'm in love with the English language. So we coined the term pyroptosis from the Greek roots pyro relating to fire or fever, and tosis to denote a falling, to describe pro-inflammatory programmed cell death. And this was the process for which we ascribed this alarm ringing, screaming process whereby we could recruit other functions to a site of danger. So pictorially, enter the scientist Susan Leilani Fink. We know that these microorganisms associate with a phagocyte. This protease, caspase-1, is activated. It leads to the cleavage of DNA. This is a very bad thing for a cell if a cell wants to divide. So this cell is functionally no longer going to live. Downstream of this process, stuff happened. The cells lysed, and they released their intracellular contents. So the first hypothesis was that host cell lysis and possibly cytokine release would depend upon disruptions of the host membrane. And so to study this, we used fluorescence microscopy, and we asked questions about cells that you can see here. These are phagocytes. They're stained blue. In this condition, in phosphate buffered saline, almost all the cells are perfectly happy. And then we challenged these cells to see if they could keep out dyes. One, I call little red, is able to go through small breaches in the membrane. Big red requires larger breaches in the membrane in order to penetrate these cells. So if we do a control experiment and we subject these cells to detergent, causing breaches in their membrane, we can say that they take up little red and they also take up big red. And when we challenge these cells with a pathogen that can cause inflammation, we see the following. Here you can see that the phagocytes are challenged with bacteria that in this slide are green. And you can see that some of the cells that are interacting with the pathogen now take up little red, but they do not take up big red. And this therefore suggested that discrete membrane lesions form during this process of pyroptosis. What is very important about this, and the reason I show you the data, is that this uptake of little red can be blocked if we block the activity of that cysteine protease called caspase-1. And so what this indicates then is that the breakdown of the membrane of the host cell is driven by a host cell process. It's not because the bacterium is poking holes in the membrane. It's because the host cell is actually biochemically capable of causing membrane breakdown. And this, most importantly, indicates that these defects result from host functions. That suggests then that in addition to DNA cleavage, that downstream of the protease, a biochemical pathway is elicited that leads to discrete membrane lesions. Stuff happens, the cells lyse, and intracellular contents are released. Let's summarize some of Susan's other more important observations. She also was able to show that these cytokines can be released, and in their active form, they're pro-inflammatory. So this is step one in causing inflammation, is the release of active cytokines. Importantly, these membrane lesions that form cause pathological ion fluxes that lead to water to come into the cell. And the cell then begins to swell. And you'll remember a classic sign of inflammation, tumor or swelling. And upon swelling, this eventually leads to lysis of the cell, and we get the double whammy for inflammation, which is the release of intracellular contents. And so a process, a host cell process, that leads to protective inflammatory responses can be triggered by the activation of this cysteine protease that leads to the release of active cytokines and the release of intracellular contents, which are inflammatory and begin the alarm to sound. Part 3. We've just described a process where a cysteine protease, caspase-1, can lead to the cleavage and elaboration of inflammatory mediators. The question then is, there more than one way to die? For you and me, certainly. And I would ask you, do you care? And I would say, I bet you do. Being run over by a truck at the age of 25 is very different than dying of old age. And one of the reasons that we might care about different pathways of cell death is that we need different functions to be carried out wherein we remove unwanted cells. Inflammation is the process of removing an unwanted cell that's dangerous to the surrounding tissue, the organ that is composed of that tissue, and the organism that has that organ. There's another process that is non-inflammatory, and it is called apoptosis. And what is important is that it's also mediated by these cysteine proteases. They're also called caspases. They're from the same family, but functionally they're very different. I would point out to you, for example, my daughter, her last name is Cookson. To her great benefit, she swims fast, she does flip turn, she continues to swim fast. I'm also Cookson. I swim, not fast, can't do flip turn. Keep swimming, not fast. So they have the same name, but they have very different functions. And the end product of this pathway of cell death is that it's non-inflammatory. And so why is this important? This term was used originally by Hippocrates thousands of years ago. And it's Greek to describe dropping off or falling off of petals from flowers and leaves from trees. And importantly, it's pronounced ptosis with a silent P. And the reason this process is important, these were a group of pathologists that made very important discoveries by looking at tissue sections. And apoptosis is well suited to a role in tissue homeostasis. It can result in extensive deletion of cells with little tissue disruption, and there is no inflammation. You took a shower today, many of you. Thank you. You washed away hundreds, if not millions, of cells that had died, and you did not suffer inflammation. In utero, okay, back when life was as good as it could ever be, in utero, everything was taken care of. Trash automatically taken out. Nutrients automatically provided. During embryogenesis, you had unwanted cells, but you did not want inflammation. So it's important to have a non-inflammatory process to remove cells, just as it is important to have inflammatory processes to remove cells. So there are two distinct cell death processes, one that's inflammatory and one that's non-inflammatory. Enter Tessa Bergsbakken and a different infectious model. Yersinia. Yersinia pestis, the causative agent of plague, has caused multiple pandemics. Epidemics, that's peanut butter coming from Minnesota. That is jalapeno or habaneros coming from contaminated farms somewhere south of the border. Pandemics means worldwide death and destruction. Okay, you may be familiar with Monty Python's Bring Out Your Dead. That's a reference to the Black Death or plague. And so one of the pathognomonic signs of plague is the bubo. It's a very inflamed lymph node. There are close cousins to pestis, and they cause gastroenteritis as well as lymphadenitis. Gastroenteritis, the gastrointestinal tract, inflammation. Lymphadenitis, that's inflammation of lymph nodes, as well as systemic infection. And so common features of these organisms is that remarkably, they fly right into the jaws of our immune system, that is lymphoid tissues. They love to grow there. They have a set of genetic attributes encoded on an entity we call a plasmid, and these organisms are all capable of causing phagocyte cell death. And their abilities to do so are encoded upon this plasmid. Going to the library is extremely useful. But I was surprised when we learned that Yersinia, the etiologic agents that cause all this inflammation, were capable of causing non-inflammatory cell death. An important observation in the literature, however, was that this process occurs in naive phagocytes. Naive meaning they're resting. Naive meaning that they're not angry. Naive meaning that they have not received danger signals. And so the question was, what might happen to activated phagocytes? The reason we thought that was an important question to address is because phagocyte activation and cytokines that cause phagocytes to become angry are important for resistance to this type of infection. The other reasons we thought it was important was because multiple activating ligands would be present during infection in vivo. So what's a ligand? A ligand is a bacterial product that is recognized by a host cell. And a bacterium by definition is foreign. And we have evolved a system to distinguish foreign from self. And so bacterial products or ligands bind to receptors or catcher's mitts on phagocytic cells. And that causes them to become activated and to be angry. We also knew from the literature that activation of phagocytes alters their responses to stimuli that cause cell death. And most importantly, as a clinical microbiologist, there was a biphasic host response associated with Yersinia infection. Interestingly, despite the fact that the microorganisms choose to grow right in the midst of our immune cells in a lymphoid organ called a lymph node, the initial rounds of bacterial replication occur without any inflammation. In 24 to 48 hours, inflammation begins. So our hypothesis is that upon activation or the receipt of danger signals, phagocytes that were infected might be redirected to undergo a process of inflammatory cell death or pyroptosis. And that turns out to be true. And shown here are uninfected cells. Actin is a major cytoskeletal element of these cells, so you can appreciate a cell there and there. In this panel, you can see cells that have been infected with bacteria, and you can see them because they're red. That shows where Yersinia is. But also, this panel is stained for the activation of the cysteine protease, caspase 1, the protein that cleaves other proteins, the protein that cleaves IL-1 beta and triggers inflammation. And you can see that Yersinia is capable of activating this process and, importantly, causing the maturation of cytokine into its biologically active form that's capable of causing inflammation. So, an organism that can initially grow without inflammation is ultimately tripped up by our immune system that is then capable of generating an inflammatory response. So we viewed it to occur like this, that a naive phagocyte initially encountering Yersinia bacteria causes the activation of a certain cysteine protease that leads to non-inflammatory cell death. However, as infection progresses and more bacteria arrive, receptors on the surface of these phagocytes, remember receptors are the catcher's mitts that are binding to bacterial products or ligands. And so that when these cells become activated by binding bacterial products, then the process of cell death is switched to something that's host beneficial or inflammation. And now I'd like to show you some data acquired by the lovely and talented Wendy Loomis from our lab. And the cell to watch in the upcoming movie is this cell, because you're going to see that it's going to reach out over time and interface with some bacteria. And then, unfortunately for this phagocyte, it's going to expire. And the way we know that it expires is because we've labeled it with this glow-in-the-dark green protein. So that's going to be shown in this next little movie. Watch the macrophage right here. You can see it reaches out, it grabs, and boom, lights are out. So what have we discussed so far? We discussed one toxic stimulus, Salmonella, that is capable of activating this pathway of cell death. But importantly, a completely different stimulus has sort of a stealth approach in that it tries to grow in the host without causing inflammation. And the very most important finding that I emphasize to you is that through activation, through the detection of danger signals, the host redirects the process of cell death away from non-inflammatory cell death to a process that is host beneficial or inflammation. So pictorially represented, this puts into context the idea of medicine and poison differing only in the dose. And that is that quiescent cells can undergo an inflammatory process that leads to inflammation. But more importantly, when these cells become activated, they're more susceptible to this process. And we believe that this is the dominant process that occurs in vivo. Because as these cells undergo inflammation, they release products that activate quiescent cells, and so that a larger population of activated cells now undergoes inflammation, and we have a positive feed-forward cycle here. And so this is of great benefit. As there are increasing numbers of pathogens, you have increasing doses of inflammation to fight those pathogens. And with regard to infection, we ultimately hope that there's resolution, because this process that we've outlined by studying microbes is actually the process that's essential for vaccines that work. They promote this process of inflammation and cause the triggering of another type of immune response which has memory. Also important is that here we can find pathological conditions where the feedback loop doesn't stop and it persists. And so here we get the generation of chronic inflammation. Chronic inflammation often is the harbinger of tissue damage. And under the right circumstances, chronic inflammation is the harbinger of malignancy. Because in the presence of inflammation, cells often do not necessarily respond appropriately. And so it is here, being stuck in this feed-forward cycle, that the poison is evoked for an organism. Part 4. So we've discussed then now that microbiology is the career of choice because an inflammatory system that has evolved over millions of years can be probed by studying the interaction of the host cell with the microbe. And that microbes are capable of triggering a response in the host that leads to inflammation that is beneficial to us. Importantly, we've also learned that cell death can be redirected. And in that research, it holds promise that we will be able to, at some point, understand the cause, understand the mechanism, and be able to redirect cell death so that we can modulate or ameliorate or abrogate important processes like heart attack and stroke and cancer. And so the biological implications for pyroptosis are diverse in that it's been shown to occur in response to a lot of bacterial infections, viral infections, but also heart attacks, stroke, and malignancy. And so the hypothesis that Susan and Tessa investigated was, is it possible that diverse signaling mechanisms or stimuli trigger a common downstream caspase effector that leads to cell death? i.e., is this process limited to infection of a certain type of organism, or are there very different biochemical upstream pathways that may converge on a single effector pathway that is critical for the development of inflammation? So as opposed to the single stimulus that I discussed earlier, or the Salmonella bacteria, is there a diversity of toxic stimuli that are capable of evoking this particular pathway? And one important experiment, I think, illustrates this nicely. Here you can see resting cells stained here with blue, and in the presence of phosphate-buffered saline in an uninfected state, you do not see the activation of this cysteine protease. There's no caspase-1. When you infect these cells with Salmonella, as we described previously, you can see, for example, the bacterium staining blue here and here causes activation of caspase-1 not only in the cytoplasm of these cells, but also in a concentrated focus in what we have termed the inflammasome or inflammasome complex. We also used a completely different stimulus, and that is the lethal toxin, a purified recombinant toxin from the etiologic agent of anthrax, Bacillus anthracis. And you can see here that you get the same subcellular activation of caspase-1. And so to summarize lots of work, what we found is that caspase-1 activation can occur through a complex pathway that requires the toxin to be released into the cytoplasm where it can do its damage. And in the presence of an adapter molecule coded here as a blue circle and called NALP1, caspase-1 is activated. Now, the Salmonella bacterium takes a very different path to cause caspase-1 activation, including a completely different activator or adapter molecule coded here green and called IPAF. But it leads to caspase-1 activation. And what's important is that irrespective of the pathway that leads to the activation of this protease, the downstream steps are as we've described earlier this evening, which is the host cell destroys its DNA, it liberates active inflammatory cytokines, and it effectively ruptures the cell, which is, from a defense standpoint, removing the niche where bacteria can replicate. So Salmonella is a pathogen, as we described, that depends on phagocytes. By destroying that phagocyte, you remove the space in which the bacterium can grow and live. And so this swelling and release of intracellular contents provides the double whammy of inflammation. So what we've discussed this evening is that multiple toxic stimuli converge upon a conserved pathway of pyroptosis that leads to inflammation. And importantly, stealth stimuli or stimuli that can initially trick our immune system, often this process can be redirected to a host beneficial inflammatory process that we've called pyroptosis. So I close then by acknowledging the other members of my group, both past and present. I didn't get to talk about everyone's work this evening. I'd also like to recognize your tax dollars, funding the National Institutes of Health, the National Institute of Allergy and Infectious Diseases, and the National Human Genome Research Institute. I'd also like to thank my important collaborators in the Northwest Regional Center for Excellence that's headed by Sam Miller, and also the Microscale Life Science Center that's a collaboration with a number of University of Washington engineers and the Dean of the School of Engineering at Arizona State University. And if you'd like to learn more about this process, I point you to this publication in the 2009 edition of Nature Reviews Microbiology. Thank you for sharing your evening with me. I'd be pleased to take your questions. Ma'am?
So is there any evidence yet that non-infectious inflammation involves caspase-1?
That's a wonderful question, and I will repeat the question. Is there evidence that non-infectious inflammation involves caspase-1? And the answer is yes. So in that cycle that I showed wherein there's a positive feedback loop. One of the problems with inflammation is it begets more inflammation. That's both the benefit and the liability. It is a pit bull, and the liability is that it can be rough and mean. The benefit is that it's a ferocious defender of its territory. So caspase-1 is involved, and for example, there are genetic mutations that cause disease called Mediterranean fever, and they are mutations that lead to upregulation of this caspase-1 dependent pathway.
Thank you.
Sure?
It's a question, I'm sure, born of my own ignorance, but why does the body need two processes for cell death, apoptosis and pyroptosis? And the second part of the question is, do you think there's any benefit in looking at ways to reverse the natural conversion from apoptosis to pyroptosis, so that if pyroptosis is taking too much energy out of the system, we're not providing the benefit that we want from a medicine point of view, that we might convert that process into apoptosis or regulate that process so that there's less pyroptosis in the system.
Okay, two excellent questions. The first question is, why do we need two mechanisms of cell death? And the honest truth is that we're not limited to two mechanisms of cell death. There are many mechanisms of cell death. It turns out that there can be predominant forms, and those forms might predominate depending on the specific situation. So for example, one aspect of responding to an infection is the elaboration of mature IL-1. But one can also get a response to infection that is frank necrosis of the cells, or just their breaching of their membranes and release of intracellular contents. And a situation where that might occur is when the concentration of toxic molecules is so high that rather than undergo this programmed process, it directly impacts the integrity of the membrane. And that's the beauty of the system, is that there are multiple fail-safe mechanisms for a cell in sort of an altruistic way to warn the surrounding cells that a tissue might be subjected to danger. It also turns out in non-infectious systems to have multiple cell death pathways is of host benefit as well. And so you can imagine that, for example, in response to anoxia, or what happens when an artery is clogged by a clot, either in your brain or in your heart, is that there is a gradient of response to that insult. And so that by having differential responses throughout that gradient, you can actually minimize the total amount of pathological damage that occurs in a tissue, and therefore, hopefully, for the most part, save as much of that organ as possible. And you can see the evolutionary fitness of such a complex system is that when an individual underwent such a pathological event, their fitness would be increased if they were able to minimize the damage. So it turns out that pyroptosis and apoptosis are two wonderful extremes of cell death, one that's non-inflammatory and one that's very inflammatory, but there's a spectrum of processes. Now, the second part of your question actually relates directly to the clinical applications of this research, which is it possible by understanding the mechanism of cell death that we will actually then be able to redirect it? And my sense is that is possible. And so some of the work that's ongoing in our lab now is to try to understand what specific triggers lead to one mechanism dominating over another mechanism, and really to be able to titrate that interaction. Because from a therapeutic standpoint, what's important to appreciate about our self-defense mechanism or self-defense mechanisms is that they're a series of interrelated and overlapping threshold-driven responses. So that sometimes small changes in a particular parameter can have a dramatic effect. And so that's where I think the results that I presented this evening about actually demonstrating through activation of a cell that we can redirect the process of cell death is that it cuts both ways. So you can now imagine therapeutic maneuvers where you activate cells, predisposing them to pyroptosis that allows you to avoid infection. And it turns out that that sort of Gedanken or thought experiment has been demonstrated in the literature previously, that a pre-existing viral infection, smoldering viral infection, will protect you against a bacterial infection. Now, similarly, we'd like to be able to push the system in the other direction. And that's the subject of intensive research by Big Pharma, is to be able to block the process of inflammation so that, to block the process of inflammation and hopefully redirect it into a process that is less inflammatory and going to cause less overall damage. Because once that positive feedback loop gets going, as the woman previously asked about non-infectious causes of inflammation, it's difficult to ameliorate. And that's part of the reason that anti-inflammatory compounds have been found to be particularly useful in treating lots of these diseases. Yes, ma'am.
Hi. What exactly are the adapter molecules? Are they electron transporters for the activation of the caspase-1 activation?
Okay, that's a wonderful question. So what are the adapter molecules that lead to the activation of caspase-1? So what those are a series of proteins that work together to scaffold and make that what we would call a supramolecular complex, right? Because you could see the focus of caspase-1 there. So the thought is that actually it's almost of organelle stature in its capability to activate caspase-1. So it's like a scaffolding. It brings together all the players so that then they can make music, if you will.
Thank you.
You're most welcome.
So if there are no further questions, I'd like you all to join me in thanking Brad again for the wonderful lecture, and I hope you'll come to the reception in his honor afterwards. Thank you, Brad.
Thank you very much.